Okay. So my name is Abdul Majeed. Currently, I am studying Master in Bioinformatics in Pakistan. So this is my BS project that I am going to discuss and going to present with you people. In silico mutation analysis of PHLHA9 gene causing complex camptodactyly and mesoaxial stenocytic syndactyly. So this project is held in 2020. So I was the leader of this project. Whole process was in silico on the base of bioinformatics, SNAP tools, and also we take the novel gene because haven't no one worked on this gene before us. So let's outline of my presentation as first of all I will introduce and I will do introduction of the gene, and also I will do the deep introduction of polydactyly and causes of polydactyly, type of polydactyly, and further I will discuss aim and objective of this project. And third, I will discuss tools and methodology that I use in this project. And after that, I will discuss results, that what type of result we get in this project. And after this, I will give a little bit touch to discussion, and also I will give touch to conclusion. And if someone want to work on this project and this date and silico work, then she can do. So let's do introduction. First of all, we must know about polydactyly. What is polydactyly? Polydactyly means the extra finger of hand and toe is called polydactyly. Mostly humans have 5 fingers. When there is some outgrowth beyond the thumb or little finger, this is called polydactyly. And also there are some malformation of hand, malformation of other body parts. In one situation, if this polydactyly is syndromic polydactyly, and non-syndromic polydactyly, we only development of extra and finger. So it is the deformity of hand and foot and human. So it's have various different type of polydactyly. Depends upon the individual, person to person, what type of mutation occur and what type of polydactyly, what type of disorder will become. So mostly we will discuss, we will point out that the small rise of length of soft tissue containing no bones called nevus. So mostly we have seen that few and children that have little outgrowth of fingers that have no bones. This is just like a flesh, and also a small outward of. I mean that there's a small growth of fingers. So and also it is formally also in toe, and also it contains some bones and joint. It is the complex form of the polydactyly, and sometimes it may be fully functional finger of hand or toe with tissue, complete tissue like other fingers, and complete bones like other fingers, and there will be the complete joint. If we do the X-rays analysis, we can see the bone and their joint. So in the front of you people, there's a picture of hand. The on the right side of hand, there is the extra wearing finger. It's and thumb, and also it's including little finger. So this is the simple type of polydactyly. Slowly, And gradually we will discuss subtype of polydactyly and sub branches of this polydactyly. Each and every branch have also subtype of deformities, deformation, and order. So polydactyly of hands and foot have various morphological phenotypes. Means they can be and various appearance depends upon the mutation, location of mutation, and also function of gene. As there is many type of gene that are involved in the formation of polydactyly, like I will discuss about GL1 gene. So when mutation occur in GL1 gene, so it will cause syndromic disease. Means disorder will be syndromic. It will affect your hand. It can affect your head. It can affect your ears, nose, and other part of the body. Uh, so as I discussed that polydactyly can be syndromic and non-syndromic. So next one is classification. How much types and how the polydactyly are classified. Polydactyly are classified on the basis of position of extra finger to toe or non-syndromic polydactyly is classified into three types. We are only discussing with non-syndromic polydactyly. If we discuss with syndromic polydactyly, there will be a huge amount of study behind the scene. So all these types are different from one another on the basis of finger, extra digit, extra finger, either it's on hand or it's on foot. So first one is post-axial polydactyly. Second one is pre-axial polydactyly. And third one is central polydactyly. So types of polydactyly, as I discussed it, pre, post, and central. So in the first sector, pre-axial, there is the extra finger on the thumb. Okay, at the thumb, there's an extra outgrowth from the outgrowth of finger that are like a thumb. Okay, so it is called pre-axial. Central polydactyly, if there is the extra finger, either side of ring finger or middle finger, this is called central polydactyly. It is in the most rare cases. And the third one is post-axial ulnar. It means that if some disorder and some pupils have disorder like that, the extra finger is on lateral finger of right hand or left hand, where this will be called post-axial polydactyly. So let's discuss the subtype of post-axial polydactyly. Subtype of post-axial polydactyly is type A and type B. So post-axial polydactyly type A. So this type of disorder is fully developed extra finger. Okay. Here digit means extra finger. The fully developed extra digit is articular with either developed metatarsal or metacarpal, and also it's linked with the fifth finger. It may be metatarsal or metacarpal. It means that there will be the deformation of fifth finger, sixth finger. The finger of this type will be metatarsal or metacarpal. So it is a bony finger. One or three bone elements depend upon its size, which result of flexion, wrinkle, and well-developed nail. And post-axial type A. Okay, it contains extra finger with bone. Okay, and also it contains well-developed nail. The extra digit may cause difficulty in every body life affected person or not. So it depends upon the formation of the finger. It may be difficult to take something, difficult to grab on something, difficult to touch something, means it depends upon the person's life. And also 24% of patients have type A with ulnar polydactyly that is associated with syndromic and congenital anomaly. Means 24% people have syndromic postaxial polydactyly, and the left 70% or 25%, 75% people have bilateral polydactyly. And the postaxial polydactyly subtype A is divided into further 6 types. Postaxial polydactyly type B. So now we are discussing with postaxial polydactyly type B. So it is also we can say that the extra finger is not well developed like postaxial polydactyly type B. And also it occurs in the form of skin, in the form of flesh. It haven't well developed size like finger. It haven't usually it have a small nail, okay, but no bone. The size of this type of disorder, length of this disorder finger is 2 to 3 centimeter long, and also it contains having a nail. If it can be on the left hand or and also it can be on the upper limb are mostly affected. So this type of fertility is complete show more complicated genetics, and thus this estimate percentage as about 43%. So it is the more complex form of polydactyly, postaxial polydactyly, than polydactyly type A, because 43% patients have this disorder. And also it involves in the non-functional text and bigger production digit having a proximal stand, break, and deficiency bony element. So this type of disorder haven't well-developed finger, but the size of the outgrowth of finger is like a ball. We can say it's like a ball. It disturbs the person daily life. And the tissue of this finger will be more than other normal finger. So now we are discussing pre-axial polydactyly. It is radial polydactyly that are most common form of polydactyly, and the occurrence of this polydactyly is genetic population, means Africans and American people have this type of polydactyly. And the extra finger is related to anterior to the thumb. So the thumb contains extra finger, means where the normal thumb is occur, the extra thumb, small thumb are the same size, the same shape, other thumb will be outgrowth in this disorder. So this is less common in our daily life. If we discuss with Asian people, if we discuss with other race of the world, and most it is occur 1 in every 1,000 people. Means this case is 1 out of 1,000. It is reported by NIH, National Institute of Health. So this is the first factor of pre-axial polydactyly. We can see that the extra anomaly, extra digit, extra finger is attached with the thumb. And also we can see the bone formation, and also we can see the X-rays in the form of X-rays with how the extra finger can disturb our hand formation, disturb our daily routine. And the first case only outgrowth, and the third case is the more complicated, so on, so forth. And also division of second tarsal is also increases. So this type of disorder are further specified in four types. So now we are discussing with syndactyly. It is the word syndactyly means finger means together, and dactyly means digit. When two fingers come together, this will be the type of syndactyly. And also this type of disorder, this type of malformation of fingers are and different variety. It depends upon the phenotypical appearance of the finger between family. Means patient can be weird, some difficulty, and also the combination of the finger will be complete or incomplete. And also a single patient phenotype may be unilateral, it can be unilateral and bilateral, symmetrical, asymmetrical, complete, incomplete, bony, and also involving in many distant bones with interland phenotypic variation. It means that the coming of this order will be unilateral or bilateral, symmetrical, and also it depends upon the family to family or patient to patient. And syndactyly and polydactyly are to the are the more frequent congenital limb abnormality. It is non-syndromic syndactyly, and also it is non-distant type. This type of disorder have further classified into 9 further groups. So now we have a picture that simple incomplete syndactyly, and also in the B picture we can see simple complete syndactyly, and third picture we can see complex syndactyly. Why it is complex? The bone of two fingers are combined. Bone of two finger, when bone of two finger are combined, the shape of the hand, the shape of the finger will be changed. And also in the most complicated form of the pic, we can watch in third D picture. It is the most complicated part in which the bone are dislocate and in irregular manner. So syndactyly are classified into further 9 types. So what is camptodactyly? It means that bent finger in Greek. Camptodactyly means bent finger means the finger that have some change in the joint. Means there will be some angular change, angular change in the joint. And which location it will be changed? It have in simple camptodactyly consists only flexion deformity of PIP means proximal IP joint. So we can see in picture that camptodactyly, the finger are bent, the lateral finger are bent, and the joint are step joint. So what is complex camptosynpolydactyly? It is autosomal recessive disorder. It can categorized by hand and foot deformalities. And also camptodactyly is a type of hand deformation that is characterized and grossly malformed hands, polydactyly with digits arising from dorsum of hand, and also thin and camptodactyly of the thumb finger, soft tissue, syndactyly of the first and second toe, and hypoplastic nails. It means that this is the complex form of camptosynpolydactyly that can be changed the whole shape, the whole facial formation of hands, and also it can be disturb our other finger, and we cannot grip things correctly, It means it can disturb this type of disorder, disturb our hand function. So we can see that how the in the middle of the three pictures, we can see that our hands are disturbed and some are bent, some are. It means that the grossly malformed hand, and also we can check the X-rays radiation, the change in the bones of the finger, metatarsal and metacarpal. And the last one is mesoaxial synosttic syndactyly. Mesoaxial syndactyly is phalangeal reduction, and also it is extremely autosomal recessive limb abnormality that is characterized by fusion of third and fourth finger. When third and fourth finger means central polydactyly, we will locate it on central polydactyly. That the fusion of third and fourth finger also uh, fuse together. The bones, the flesh of the third and fourth finger fuse together. It form a disorder which is called mesoaxial stenotic syndactyly. So it occurs mostly in the feet and also in hands. So we can look at the picture A. The X is the radiation of picture A is E. So let's see check. It is the fusion of second and third finger. So what happens? The bones become together and also disturb life. And also we can check the fusion of picture B, better feet, and also the fingers and their bones are also presented in the front of us as a X-ray radiation. And also in picture C and D, we can check their bones and also their flesh and also their tissue structure, and the scene behind the tissue structure is bone. So now let's come to the point uh, of our main gene that involved in the mesoexosyntactic syndactyly and also camptosynpolydactyly, complex camptosynpolydactyly. So function of BHLHA9. What mean by BHLHA9? Basic helix, loop helix, A family member 9. So A is the family member of this gene. So BHLHA9 gene contribute to and the basic domain DNA binding domain means this gene is functionally contain the DNA binding site, and also it can help encode protein in the transcription factor involved in the limb development. This gene is involved in the formation of limb development. And also the member of this family have two conserved domains. One is basic domain, which is bind to constant hexanucleotide sequence known as EBA. So the first one is basic domain. This is hexanucleotide sequence. Basic domain contains 6 nucleotide sequence. The 6 nucleotide sequence called EBA, and the other domain is helix loop helix. This domain contains facilitated the interaction between the other protein. The second domain have functionally interaction with other protein to perform a proper, a well-developed function. And also the domain contain homodimers and heterodimers. So homodimer means same type of sequence. Heterodimer means different type of sequence. And also this gene is involved in the regulation of molecular and cellular activity during limb development. Before birth, it involved in the molecular and cellular activity and the limb development. So what wrong in BHLHA9? So what will be the malformation of this gene? So it calls the disease. First of all, we will check the reason why this gene are involved in the disorder formation. Mutation. First point is mutation. Mutation means the sudden change in the sequence of DNA and also alter DNA binding property of BHLA9. If mutation occur in gene and this gene, so it will change the DNA binding domains of this gene. So and also due to mutation, abnormal protein formation will come out. And the output of mutation will be abnormal protein formation and also change active site of protein. If if the mutation is an active site of protein, this mutation will change the active site formation of the protein. Means the ligand, the active molecule, the like lock and key model. So a specific key can open a specific lock, or for one lock, that here there will be one type of key. Means if active site of mutation, if the mutation comes in active site, so the protein will not be functional. If it's functional, the altered, the mutated protein or abnormal protein will be formed. So BHLA9 gene were found, where the location of BHLA9 gene in human chromosome, it located, the location of BHLA9 gene is on 17 chromosome, RM is Q, position is 13, and subband is 3. So this gene contains only one exon, and also this gene is about 1372 base pair long. And if we check the molecular location of the chromosome of the sequence of this gene, start from 1270 44 4, and also the last position is 1271 81 5 base pair, and also it contains 708 bases cDNA. cDNA means the coded DNA that will produce the desired protein, the desired amino acid. So on the base of cDNA, this gene encodes 235 amino acids. And also at the UniProt ID is given in front of you, PM7 RTU4, and also the ensemble ID is in front of you people, and also the ensemble reference sequence ID, expression, we can say expression number is in front of you people, and also the disease associated with this gene is complex Camptosynpolydactyly, and also the second one is mesoaxial synostautic syndactyly. So what is the aim and objective of this project? The first one is impact of MNP on BHLA9 protein. What is the multiple nucleotide polymorphism on BHLA9 protein? So we will discuss this later, and result formation and result that what means by MNP, multiple nucleotide polymorphism, and our main objective is function. What was the normal function of this gene? After mutation, what function, what deformation, what abnormality come, and also we will discuss about the protein structure, as the protein structure is NPDB or not, FPDB, if protein structure is validated or determined or not. We will discuss it later. And also protein, we will discuss about the protein stability, And also we will discuss the functional and structural correlation of BHLHA9 protein. So tool and method. Tool and method that are used in our uh, project is Swift, Philippine 2, Provide, iMuted sort, Potham, and PDSNCs. These six different type of tool are used for single nucleotide polymorphism. If we are looking forward to the effect of the mutation, either it is harmful effect or normal or neutral. And also we use for the protein sequence structure prediction PSI, correct? And also we use for sequence structure prediction Raptor X. And also we do mutagenesis. We do mutagenesis in PAMOL. PAMOL is the visualization tool that are used for the to visualize the 3D structure. And also we can do further analysis inside the PAMOL. And our fourth one is energy minimization. We do energy minimization and PDV liver. And also fifth one, superimposition. We did the superimposition